Hi everyone. So in this lecture, we're going to talk about how we classify and identify different microorganisms, different microbes, and we're going to focus on the three domains of life. So we're going to talk about what the word taxonomy actually is, how people came up with this three-domain classification system, and if you're given a bacteria, which is a prokaryote, how do you identify what exactly it is? So for example, if someone has an infection, and you get a stomach culture sample or a urine sample, what experiments do you actually do to figure out what type of organism it is to give them the proper treatment? So we'll start off with some definitions. So taxonomy is studying organisms so that you're able to arrange them into groups. So all of taxonomy is how you basically arrange organisms into various groups. And the reason why this is important is so that we can classify or identify an organism. So think about, I know most of you want to go into the health field. If someone has an infection, before you can treat it, before you can do anything, you actually want to know what the bug actually is, what the microorganism is. And if it's something we know nothing about, like it's a new microorganism, the first thing you'd want to do is classify it. Is it a bacteria? Is it an archaea? Is it a eukaryotic organism? A eukarya, maybe like a parasite that's causing an infection? And then from there, you would want to identify specifically what it is. So if I classified something as bacteria, okay, what type of bacteria is it? Is it gram negative? Is it gram positive? Is it E. coli? Is it salmonella? Is it H. pylori? So that's the purpose of taxonomy, is to arrange organisms into groups. And it's actually the science of classifying organisms into what they are. So for some reason, this image is not showing up on here. You guys have all my slides, so you can look at them. This image was, I think, a three-domain system that you're supposed to see, and it shows you how to identify organisms. And it's important to identify organisms in medicine so we can give proper treatment. So we classify organisms into three different domains. This is what we call the three domains of life. There's domain bacteria, domain archaea, and domain eukarya. And the way that scientists decided to split all life into three domains is by sequencing something called ribosomal RNA. When you look at the ribosome, inside the ribosome, there's a little RNA molecule that we specifically call ribosomal RNA. And if you take that RNA molecule, and these days in lab, you stick it in a machine, you basically pipette it into a machine, it'll tell you the sequence of nucleotides. And remember, RNA is made up of A's, U's, G's, and C's. So you'll get this sequence. And when you compare ribosomal RNA sequences, you'll see that bacteria have their own rRNA sequences, archaea have their own rRNA sequences, and eukarya have their own unique rRNA sequences. So we can split all life into three domains. So that means that if you basically found something crawling and you didn't know what it is, I'm not going to classify it as a spider. You don't know what it is. You're not sure, is it bacteria? Is it archaea? Is it eukarya? You can isolate its ribosomal RNA, you can sequence it, and then you see which one of these groups does it match, and you know you can classify it into that domain. So that's what classification of the three domain system is based on. It's based on taking the ribosomal RNA, sequencing it, and then figuring out what group it falls on. Now, there's a lot of microbes, microorganisms in the eukarya domain, but if we focus specifically on bacteria and archaea, since they're all microorganisms, they're all very tiny, you need a microscope. Let's say you discover a microbe and you're not sure if it's bacteria or archaea. Well, if you sequence its rRNA, you can see which one it closely matches and then put that unknown organism in that domain. So that's a classification. And again, if you look at a ribosome, here's a picture of a 7DS ribosome. Remember, we see 7DS ribosomes in prokaryotic organisms. Bacteria and archaea are prokaryotes. All ribosomes, there's a large subunit and a small subunit, and they come together to make the whole subunit. Anyway, within the ribosomal and the small subunit is where we see the 16S rRNA. We talked about the S in class and how the S refers to sedimentation rate. So that's where scientists came up with this 50s, 60s, 70s ribosomal subunits. And again, we look at this sequence specifically because they rarely mutate. So you know that as the years go on, you can always reliably sequence the ribosomal RNA sequence and classify organisms as bacteria, archaea, or eukarya. Now, when we look at the domains, just as a quick review, bacteria and archaea domains are both made up of prokaryotic cells, and eukarya domain is made up of eukaryotic cells. And remember, prokaryotes have a 70S ribosome. So when we look at the bacterial and archaea ribosome, both of them have a 70S ribosome. Eukarya has an 80S ribosome. Now, for prokaryotes, within the 70S ribosome, the ribosomal RNA is specifically called 16S ribosomal RNA. And for eukaryotes, within the 80S ribosome, there is an 18S ribosomal RNA subunit. So if I ask you, where do we see 18S rRNA? I want everyone to say in eukaryotes. Where do we see 16S rRNA ribosomal subunits in prokaryotes? And that can either be bacteria or archaea. So more classification. So here we look at the taxonomic hierarchy. Okay, let me go back. So I think there's some things that are not showing on here for some reason, but you guys have the slides at home. So when we look at the taxonomic hierarchy, I just want to say one quick thing about this slide. The biggest, broadest group that you can identify something in is domains. So you start off with 3 domains of life, and then you can split things up into kingdoms, phylum, class, order, family, genus, species. So you go from very broad to very specific. So an example of a very specific organism is Escherichia coli or E. coli, where it's part of domain bacteria. The genus is Escherichia and the species is coli. So this is classification is deciding what domain do things belong in, all of these things. And then when you get down to the specific genus and species, this is basically you've identified the organism. So again, we classify something as bacteria, archaea, eukarya. You can do more classifications. And then identification is when you actually match characteristics of an unknown organism to a list of known organisms. And then from those characteristics, you can decide what it specifically is. You can identify it. So this is very important in medicine. So in clinical labs, you have someone who has a lung infection. I mean, this right now is very relevant with the coronavirus. Lung infections, pneumonia can be caused by a lot of different things. It can be caused by a lot of different bacterial species. It can be caused by different fungal species. It can be caused by different virus species, such as coronavirus and whatnot. So when someone comes to you with these symptoms, what clinical lab experiments do you do to identify it? And identification, again, is very specific. It's very important to medicine. So identification of prokaryotes, let me put this and we'll talk about it. So we're going to talk about 2 terms I want everyone to know, which is phenotype and genotype. Phenotype is when you identify a prokaryote. Let's talk about bacteria specifically. So identification of bacteria. Phenotype is when you look at physical characteristics. Anything you can see is a phenotypic characteristic. Genotypic characteristic is when you're actually looking at the DNA, the genes. So if you can see something, we consider it a phenotypic characteristic. So a physical characteristic. Microscopic morphology. This is a phenotype, a physical characteristic that you can use to identify bacteria. So you've all done microscopy work in lab. Microscopy tells you a lot of things. You can get the size of organisms. We haven't done it in lab, but with a microscope, you can measure organisms. You can get the shape. You can figure out, is your bacteria coccus? Is it rod? Is it spiral? This is all phenotypic because it's all using your eyes. And you can do differential staining. You can figure out, is your bacteria gram positive? gram-negative, you can do special stains, you can do flagella stain, you can do acid fast. These are all physical characteristics that you can use using the microscope. Another thing you can do to figure out what your bacteria specifically is you can look at colony morphology. So unique appearance and pigments. Remember in lab, we saw that some bacteria make distinct pigments. Some bacteria were red, Serratia, for example. Some bacteria are purple, some are green. So we can look at what they look like on an agar plate. That's what colony morphology is. And you can grow them on different types of media. So here's Pseudomonas aeruginosa, which causes bad lung infections, as an example of something it can do. So both microscopic morphology and colony morphology are physical characteristics. So they're phenotypes that we can do in lab to figure out what something is. So most trained scientists, for example, if they grow Pseudomonas on an agar plate, if you had someone come to you with an infection and you streak it, you can figure out without much more work that it's Pseudomonas because your eye has been trained to know this is what Pseudomonas looks like, this is the color it makes, all of that. So phenotype, another thing you could do to identify bacteria is metabolic testing. When we did metabolic testing, remember, you look at tubes, you see, did a color change happen? Is there a clearing on the plate? So it's different types of tests that you can see to see whether enzymes are present or not. And remember, this is all things that you can judge with your eyes. So we can look at enzymes, groups of enzymes, and we use all these metabolic characteristics to create what we call a dichotomous key. And I don't know why the pictures are not showing up, but again, you guys have the lab so you can see the pictures. So a dichotomous key is where you use the list of all the tests you've done. So gram, stain, syn test, carbohydrate, fermentation, all these tests, and you have them lead you to one specific organism. And that's what people in clinical labs do. Now we're going to talk about genotype identification. So in the previous slides, what we talked about, everything was phenotypic identification. So microscopic morphology, colony morphology, and metabolic tests are all these things that you can do to identify a bacteria phenotypically by looking at its physical characteristics. Something that you could do that's also really easy is you can identify bacteria by looking at its genotype. So genotypic characteristics are basically looking at the DNA, because remember, genes are DNA and DNA. So if I say, anytime you see this GEN, it means gene, means DNA. You can also send off an unknown organism for sequencing. So if you've got some sample, someone has a lung infection, you got a sample, maybe you didn't want to do any of the phenotypic tests, you didn't want to do the microscope work, the colony, grow it on an agar plate, you didn't want to do metabolic testing, you can just send it off for DNA sequencing, and that's looking at genotypic characteristics. And when you send it off for sequencing, you can look at its specific sequence and compare it to what's known and decide what organism it is. So sequencing DNA or ribosomal RNA. Sequencing DNA will tell you what it specifically is, that organism. Sequencing ribosomal RNA, you can use it to identify is something bacteria, archaea, eukarya, and you can use it to identify things more specifically. DNA fingerprinting. So in the biotechnology lecture, we learned about restriction fragment length polymorphisms, or RFLP, and the polymerase chain reaction, or PCR. These are two different methods where you actually look at the DNA, you look at the gene, and you use, for PCR, you use probes, remember, that bind your genes. Go back and watch that video if you don't remember. So that can tell you what your specific organism is, because if you can amplify it in PCR, that gene, it means that a gene is present, which you can use to identify the organism. Restriction fragment length polymorphism is when you use restriction enzymes that cut DNA, and then you line all the DNA fragments and you compare them to known DNA fragments from different organisms to decide what organism you have. And again, DNA probes are when you design these molecules that are complementary to a DNA sequence, And if they hybridize or bind to it, you know that you have that specific DNA sequence, which ultimately matches a known organism so that you can identify it. So we see that in colony blotting. And I didn't really talk about colony blotting in biotechnology because no one does it anymore. So you can kind of forget this part. DNA probes are used in fish. This is the fluorescence experiments where you design DNA probes that every time they bind a gene or a piece of DNA of interest, you get fluorescence. So look at the study questions and the practice tests that we have. So for example, in this study question, define taxonomy. So it's the science of classifying organisms. Which component is emphasized in medicine? That is identification part. Understand the purpose of taxonomic hierarchy and no selected components. So the purpose of taxonomic hierarchy is to start from very broad classification of organisms to very specific. So you start from domain and you go into all the different hierarchy of taxonomy to end with the specific species. Define a prokaryotic species. So remember that bacteria and archaea are prokaryotes. Therefore, they have a 70S ribosome. In the 70S ribosome, we have 16S rRNA. And then with prokaryotes, we do not have the nucleus, just as a reminder. And discuss the origin of the 3-domain classification system. It originated by sequencing ribosomal RNA sequences. Explain the different methods to identify prokaryotes using phenotype and genotype. If you have an unknown bacteria, you can use phenotypic characteristics such as microscopic morphology. You can look at the shape, you can look at the gram stain, you can look at the size. You can do colony morphology, you can look at the pigment or what the colonies look like. Are they round? Are they fuzzy? And then for genotypic characteristics, you can sequence the DNA. That's probably the easiest thing I would do. You could do PCR, you could do restriction, fragment length, polymorphisms. So all these things could be done. This is a relatively short lecture, so look at the study questions in the end on the lecture posted online. And if you guys have any questions, you know where to reach me.